Okay, let's try that again. The nitrogen cycle. There are some bacteria that can do something called nitrogen fixation. Okay, and you're going to read about that here in your chapter if you haven't already. It's the conversion of atmospheric nitrogen, so nitrogen that's in the air that we breathe, and they turn it into a nitrogen-containing molecule that organisms can use, like plants or whatever it may be. So, you'll read it, but I'm just going to cover a little bit of it. There's a specific bacteria, Rhizobium. It's on the roots of like legumes, which is kind of like a bean, and they have a special symbiotic relationship. The bacteria is protected by the roots of the plant, and then in return, the bacteria converts nitrogen in the atmosphere because it's a nitrogen fixing bacteria, and it produces ammonia. Okay, which some plants can use directly. But then there's another type of bacteria that can live in the soil that it takes that ammonia that the one bacteria turns in, takes nitrogen and makes ammonia. And then it takes that ammonia and it converts it into nitrite. And then there's a third type of bacteria that can convert that nitrite into nitrate. Thus, the production of nitrate requires 3 different bacteria. Okay, and so it's taken multiple bacteria to turn nitrogen from the air into ammonia, and then another bacteria to take ammonia and turn it into nitrate, and then there's another bacteria that will take that nitrate and turn it into nitrite and turn it into nitrate. But the point of all that is it takes 3 different bacteria to do that, to do this nitrogen fixation, to fix and put nitrogen into the environment so organisms can use it. So it just shows how important bacteria really can be, because nitrogen is very important for a lot of your plants, so plants will grow, so they can do their jobs in the world, and so we can eat them and have the oxygen they provide, and the whole cycle. All of that has to be made by bacteria, which is really, really cool. But you will need to know that for your test, that there are bacteria that take the nitrogen in the air to turn it into ammonia, and then turn that into nitrite, and then turn that into nitrate. That it's a whole nitrogen cycle. You do need to understand that. So if it's confusing, read over it a few times in your book till you get it wrapped around your head, okay? Okay, so those are the different jobs that bacteria can do. And then the book further just breaks them down, bacteria, into 3 major groups, okay? You have proteobacteria, which is gram-negative. That's why I wrote it in red, because red is gram-negative, okay? And then there's non-proteobacteria, which is gram-negative, okay? So there's your 2 major groups of bacteria that fall into gram-negatives, okay? So what's the opposite of gram-negative? Obviously, gram-positive. And then there's different types of gram-positive bacteria. They have broken those down into 2 groups. Actinobacteria and firmicutes. No. Okay. Okay, so let me pause this. I wish I could pause it. Okay, so those are the 2 further broken down into that. And then the last thing that your chapter covers, and we're going to go through a little deeper, I'm just hitting the highlights right now, is viruses, which are not living, okay? We talk about them like they're alive, but they really don't have all the characteristics of life, okay? They have to infect a living host to replicate, all right? And all these other things, bacteria, archaea, all the things that we've learned about, they have different ways to replicate on their own, right? Binary fission, budding, all these different things, but they can replicate on their own. Viruses cannot, okay? They have to have a living host to replicate for them. And so viruses is when it's all multiple virons put together, okay? Viron is the individual particle of the virus, and then the virus is when there's lots of virons put together. And then we're going to hit the tip, graze over the lytic and the lysogenic cycle, which is important. And then just how viruses work. They have to attach to the host, then they have to penetrate and inject into the host. They have to inject their nucleic acids into the host. And then now that they're inside of the cells of the host, and we've learned about how cells replicate and mitosis and all that. And so then their cells will start to replicate. So the virus starts to replicate. And then it has to mature. And then it will release from the cell if it's in the lytic cycle. If it's in the lysogenic, it will not. If it's in the lytic, it will actually release from that cell. And then all those new viruses that have been replicated will go spread to other cells inside of that host, if that's you or your dog or the plant or whatever it is. And then it continues that cycle over and over. Okay, so that's that. Let me pause this.